First of all, the two major sites of glucose production are liver and to a lesser extent kidney tissues. Because liver tissue is a major site of glucose production, let's take hepatocyte. To explain why stimulation of gluconeogenesis decreases the activity of the Krebs cycle, we have to know how cells, including hepatocyte, produce energy. Recall that hepatocytes in liver tissue generate energy by degradation of acetyl-CoA in the Krebs cycle and subsequently an electron transport chain that results in energy production. So initially acetyl-CoA is degraded in the Krebs cycle. Krebs cycle occurs in the mitochondria. Overall, Krebs cycle has 8 intermediate metabolites, and we have to know that acetyl-CoA enters into the Krebs cycle through reaction with oxaloacetate that results in formation of a citrate. So oxaloacetate is one of the crucial intermediate metabolites of the Krebs cycle, because without oxaloacetate, acetyl-CoA cannot even enter into the Krebs cycle. And during degradation of acetyl-CoA in the Krebs cycle, NAD and FAD electron transporters block out electrons out of acetyl-CoA that results in formation of NADH and FADH. And NADH and FADH electron transporters deliver electrons to electron transport chain that results in energy production. Now, how glucose is formed? In gluconeogenesis, glucose is produced from pyruvate. Pyruvate from the cytosol is transported into the mitochondria, where pyruvate carboxylase converts pyruvate into oxaloacetate. And then oxaloacetate is transported by mallet shuttle into the cytosol, where it can be used for glucose production by conversion to phosphoenol pyruvate. Then phosphoenol pyruvate, through 3 consecutive reactions, is converted into glyceraldehyde 3-phosphate. From glyceraldehyde 3-phosphate and dihydroxyacetone phosphate produce fructose 1,6-bisphosphate, then fructose bisphosphatase 1, cleave phosphate group, and fructose 6-phosphate released. Then fructose 6-phosphate converted initially into glucose 6-phosphate, and then phosphate group is cleaved, and thereby glucose is formed. Very important that gluconeogenesis is controlled by insulin and glucagon. Glucagon stimulates FBPase1, thereby glucagon stimulates gluconeogenesis, and insulin inhibits FBPase1, thereby insulin inhibits gluconeogenesis. So that's how glucose is produced. And now we see that Krebs cycle and gluconeogenesis have one metabolite in common. It's oxaloacetate. Moreover, at this point of gluconeogenesis, oxaloacetate is located in mitochondrial matrix, where also Krebs cycle occurs. So oxaloacetate can be used in both processes. And in condition as starving or in diabetes type 1, glucagon level is very high and insulin level is low. This proglucagon state markedly stimulates gluconeogenesis, and basically gluconeogenesis becomes the first priority, and this makes sense because glucose is the main fuel for the tissues. Moreover, for erythrocytes, glucose is the only metabolite that can be used to maintain their cellular integrity. Without glucose, erythrocytes simply will be destroyed. So the first priority is to maintain blood glucose level. And in starving, proglucagon state basically shifts all metabolites into gluconeogenesis, including oxaloacetate. And eventually, such strong stimulation of gluconeogenesis depletes oxaloacetate stores. And because oxaloacetate is the intermediate metabolite of the Krebs cycle, decrease in oxaloacetate causes decrease in the rate of the entire Krebs cycle. So it's the mechanism how proglucagon stimulation of gluconeogenesis at some point decreases the rate of the Krebs cycle.